Welcome, students. Today we move to Chapter 2, where we explore the very foundations of life, molecular biology and its profound implications for our world. To understand biotechnology, we must first understand the molecules that drive it. We will look at DNA, RNA, and proteins, the trio that forms the core of biological information flow. Think of it as a sophisticated manufacturing system. In this system, DNA is our permanent master blueprint. RNA is the temporary messenger carrying specific instructions. And proteins? They are the working machines that actually build and maintain the organism. DNA, or deoxyribonucleic acid, is the archive. It is sequestered in the cell nucleus, protected like a master cookbook in a library. It stores every instruction needed to build you, from the color of your eyes to the structure of your heart. Chemically, DNA is a beautiful double helix made of 4 letters, A, T, G, and C. These nucleotides are arranged in a specific sequence, forming the genetic code that defines every living creature on Earth. Because the DNA blueprint is too precious to leave the nucleus, the cell makes a copy. This is RNA. Ribonucleic acid acts as a portable recipe card that moves to the cell's protein-making factories. RNA is single-stranded and uses uracil instead of thymine. It is shorter and more flexible than DNA, allowing it to act as the perfect intermediate messenger in the flow of genetic information. Finally, we have proteins. If DNA is the plan and RNA is the memo, proteins are the actual workers. They fold into complex shapes that allow them to perform thousands of different tasks in the cell. Consider hemoglobin, which carries oxygen in your blood, or insulin, which regulates your blood sugar. These are precise molecular machines built according to the instructions encoded in your DNA. The relationship between these three is governed by the central dogma. This is the grand principle that information flows in one direction, from DNA to RNA, and finally to protein. First, transcription copies DNA into RNA. Then translation reads that RNA to build a protein. This elegant process is happening right now in every cell of your body, millions of times per second. But what happens if the instructions are wrong? This is what we call a mutation. A single change in the DNA sequence can lead to a faulty protein, which is the basis of many genetic diseases. By understanding this flow, we can perform genetic diagnosis. We can identify mutations before they cause harm and develop targeted treatments, a field we call personalized medicine. With such power comes great responsibility. As biotechnology becomes more accessible, we must address critical issues of biosafety, biosecurity, and genetic privacy. Biosafety ensures our labs don't accidentally release pathogens. Biosecurity protects against intentional misuse. We must also protect the genetic privacy of individuals as DNA sequencing becomes a common tool. Finally, the economic impact is staggering. Molecular biology is now a multi-trillion-dollar industry, driving innovation in healthcare, agriculture, and environmental sustainability across the globe. In summary, we have explored the master blueprint of life and how its information is expressed and protected. Thank you for your commitment to this study. I will see you in Chapter 3.